So I was just mentioning constitutive promoters, and something I wanted to bring up in general is that we've been talking about everything as if it's always on, and this idea too of burden, metabolic burden, is one that really motivates the case for an inducible promoter. And in the remainder of this lecture, we will dive into many other reasons why we may want inducible promoters in a different sense as biosensors. But here, I wanted to talk for a moment just to take a step back and remind everyone that we've been operating from the idea that your gene is always on. It's got this promoter, it's got this RBS, and that would be constitutive, where the promoter activity is just dependent on the availability of the RNA polymerase holoenzyme, which we talked about last time, but where transcription factors don't play a role. And so in many cases, industrially, for example, if you got a strain where you've got a pathway, or at least a core set of pathway genes that are not going to be too toxic, you don't need them super strongly overexpressed, but you want them on for production of your product, you might choose to make those promoters constitutive. Because the other reason why you might do that is so that you don't have to add some expensive chemical like an inducer to turn them on. And thinking about, a metabolic engineering process that may be 10,000 liters or 100,000 liters large, you don't want to have to add that cost and that molecule, and then you have to worry about mass transfer and all of that. At the same time, particularly if you're at lab scale, you know that a lot of your pathways are not what the cell wants to do. And so if you want to accelerate the design, build, test, learn cycle that we have in research, you want to be able to just flip a switch and maybe have your organism go from its normal growth rate to this slower growth rate where it's really focusing on metabolite overproduction. Or similarly, you see this in protein overexpression all the time, is that if you want a ton of protein, well, you use an inducible promoter. And so you can give it a really strong expression strength, but you can wait until the cells are at mid or late exponential, and then add your inducer. So we're going to briefly go over some inducers here, and then we'll dive more into biosensors and transcription factors, which are all related. So the examples that we're going to talk about are IPTG, L-arabinose, ATC, as they're really common. We've already spoken about IPTG indirectly in that we've talked about the lac operon. And so, lac operon is named for lactose, and it involves the breakdown of lactose, which is a disaccharide. And there's also a molecule that's a mimic of lactose called allolactose that can also trigger transcription of the lac operon. And IPTG is effectively a mimic of allolactose. And while these chemical structures aren't drawn in exactly the same way, you can kind of see the oxygen replaced here by a sulfur, and then this sort of branched group of carbons instead of a second sugar. Right, and so when we use IPTG for protein overexpression, we're actually doing one thing that's different than the lac operon. We are typically working with the region of the promoter and operator that is now independent of catabolite repression. We typically don't want the presence of glucose to shut down the effectiveness of IPTG. Although you will find a number of variations on IPTG-based inducible promoters, most are just focused on the negative regulation. And then effectively you substitute your genes of interest rather than the lac genes here. So very similar strategy But something I wanted to mention that's a little different here is that, arabinose is another sugar. And so this is a natural operon that occurs in E. coli. They're the ARA BA, and this should be the ARA D gene. So this promoter is named, is sometimes called the ARA-BAD or arabinose promoter, in part because the genes are BAD. And arabinose, the form that's recognized by cells, like other sugars, has to do with the chirality. It would be the D enantiomer. So you can use the L enantiomer as an inducer. And part of the reason why you'd want to do that, and I forgot to mention this about IPDG as well, is because you don't want your inducer to actually get broken down. So you can have irregular E. coli strains, and maybe some have this native operon deleted so that there's nothing going on, no response in the cell. But regardless, when you add L-arabinose, even if these genes are made, if these genes are expressed and these proteins are made, they're not going to break down your L-arabinose. Another inducer has to do with what was an antibiotic, tetracycline. Still is an antibiotic, but tetracycline is interesting because you have in synthetic biology parlance, you have two different kinds of TET bars. You have a TET repressor, and then you also have a tetracycline resistance marker. Basically what nature evolved, what drug-resistant bacteria evolved to break down tetracycline. But that is, I think, natively under the control of a TET repressor and an associated promoter. And so you can provide cells with a non-antibiotic version of tetracycline. It's just the anhydro. So it's missing, the structure looks a little different. It should be missing a hydroxyl group, which it looks to be, but is different in other ways, and hopefully just resonance. Okay, so right, you can use these three molecules. These are very common inducers, and there are more. Part of the reason why these three have had such high adoption rates is because of the characterization of these tools, as well as the strength of expression. And another thing that we want to think about is leakiness. If you're looking for that kind of inducible response, then you're probably doing it because you don't want the cell to be making this protein all the time at some basal level. But all of these systems have, to some degree, a little bit of proteins, of gene expression that's still happening, even when the inducer isn't around. And so next time, according to this slide, we'll cover transcription factors, biosensor engineering, and synthetic gene circuit design.